So again, the reaction that we took a look at in our lab was one that involved this enzyme, catechol oxidase, which is naturally found in potatoes and bananas and other fruits. And what this enzyme does, like all enzymes, is that it speeds up a chemical reaction. And the particular chemical reaction that this enzyme speeds up is one that involves a molecule called catechol and actually oxygen. And so this catechol, because this is the molecule that can specifically fit into the active site of catechol oxidase, this molecule is considered to be the substrate for catechol oxidase. And so what occurs whenever these two are able to interact with one another is that it speeds up, again, a chemical reaction, one that involves converting catechol into a different molecule, benzoquinone, and actually water is another product. And so in lab, we measured how much benzoquinone was being produced by using the spectrophotometer, which allowed us to put a number on or quantify how brown or how dark our solution was getting. And so basically the goal of our experiment was to try to figure out the condition at which our enzyme worked best at. Right? So we subjected it to different pHs, and we were interested in finding out which one of those pHs was optimal for our enzyme. We also subjected it to different temperatures, and we were interested in finding out which temperature it worked best at. And we also threw different concentrations of substrate at it, and we were interested in seeing which one of those substrate concentrations produced the most product in a certain amount of time. So to be able then to make conclusions off of all of your data, it'll first help to know a few basic things about enzymes and probably just molecules in general. And so we'll point those out here. So again, one of the things that you were interested in looking at was how different temperatures affect the activity of our enzyme. And so in order to make conclusions based off of this, it'll first help to know just this one really basic idea. And that basic idea is that whenever the temperature increases, it causes molecules within that environment to start moving a little bit faster. Right? At colder temperatures, the molecules move slower. And so if you then apply that to our situation, where we were looking at a chemical reaction involving catechol, and also we threw the enzyme catechol oxidase in there with it, if the temperature is really cold, then that means that all of these molecules you see on the screen here are moving really, really slow. And so think about then how really slow moving molecules would affect the chances of a substrate getting into contact with an enzyme in order to produce product. Right? If you end up raising the temperature a little bit, like we mentioned, raising the temperature causes molecules within that environment to just start moving a little bit faster, like this. And so the thing to think about is, well, if things are moving a little bit faster, how does that affect the chances that one of these substrates will bind to one of these enzymes? So that's one thing to think about when you're considering how temperature affects the ability of an enzyme to convert a substrate into a product. Another thing to consider, and it's a thing that you were asked to think about actually on your pre-lab assignment, is, well, we mentioned that raising it causes molecules to move a little bit faster. What if you raise the temperature a lot, to maybe even an extremely hot temperature? How would that affect the ability of an enzyme to do its job? Well, we mentioned, we've shown you up to this point that catechol oxidase basically has the shape of a cartoony Pac-Man looking thing. But in real life, that's not the case. So in real life, all enzymes are proteins. And all proteins are made up of a bunch of amino acids that are strung together and then folded in a particular way that lets it perform its specific function. Right? So the same thing with catechol oxidase. In real life, it doesn't look like a Pac-Man. In real life, it's a big string of amino acids that's folded in a particular way. And so then how would, a really hot temperature affect the ability of this enzyme to do its job? We mentioned that if you increase the temperature, it causes molecules to move a little bit faster. But if the temperature is really, really hot, potentially that could cause basically the molecule to move really, really fast, so fast that it might break the bonds that are holding together that folded protein, so that the protein ends up unraveling basically. And so in class you probably learned that, the technical term used to describe this unraveling of proteins, it's called a denaturation, right? So a really hot temperature, you would expect it to end up denaturing a protein, kind of like this. And so the thing to think about then is, well, if the temperature is really hot and it causes the enzyme to become denatured, how would that affect the ability of this enzyme to produce product? How would that affect this enzyme's ability to speed up a chemical reaction? So you're going to be asked to then visualize the temperature at which this enzyme worked best at. And the way to visualize that is to draw a graph. And the type of graph you're going to end up drawing is probably going to look really similar to this. You're going to be asked to plot the amount of product produced over time. And so say that you plot your data for one of your temperatures. So you mix together enzyme and substrate and you subject it to a particular temperature, and then you plot all your data points and you see something like this. Right? And say now that you plot your data points for one of the other conditions. So you have the same amount of enzyme, the same amount of substrate as this reaction. The only difference is that you subject it to a different temperature, and when you plot those data points, you see something like this. So basically you see that in reaction B, right? In this amount of time, it produced a much less amount of product compared to reaction A. So in other words, reaction A has a much greater reaction rate as compared to this. And so by looking at these two lines, then you can tell which particular temperature, if you knew what temperatures these were, you could tell which particular temperature was more optimal for our enzyme. Say, for example, that, we'll say now that you plot your data for one of the other temperatures, right? So you have another tube, you put the same amount of enzyme and the same amount of substrate in it that you did in these two tubes. The only difference is that it's a different temperature. And you start plotting that data and you see something like this initially. Right, so at first that might indicate that this reaction rate, it's pretty steep. So this condition looks like it's going to be a lot better one as compared to this condition or this condition for the enzyme. But what if after a relatively short amount of time you see this happen to this reaction, so that no more product is being produced after this particular point? So if you end up seeing something like this, some things to think about then is, well, what might have caused the chemical reaction to basically stop? Two possible things that could cause it to stop is, well, one, what if all of the substrate ran out? Right? If there's no more substrate for the enzyme to convert into product, then no more products will be produced. So that's one thing that could cause the chemical reaction to stop. Another thing that could cause the chemical reaction to stop is, well, what if something happened to the enzyme? What if it gets trashed for some reason? That's another thing that could cause this to happen. So if you see something like this, basically what you're going to want to think about is, what caused that to happen? Was it because the substrate ran out? Was it because the enzyme got trashed? Was it because of some other reason? And you should be able to figure that out by thinking about everything that you see on this graph and by thinking about how you set up your particular experiment.